Hi everyone, today we will study about laboratory diagnosis of viral infection. So whenever any patient goes to doctors with any symptoms or illness, initially the doctor will examine the patient and depending upon the suspected illness, he will order for different types of laboratory test. If the viral infection is suspected, he will collect the sample and send to microbiology or virology laboratory for the diagnosis. So, common types of samples for the diagnosis of viral infection involves nasopharyngeal swab, blood, body fluids like cerebrospinal fluid, urine or stool samples, scrapings, biopsy samples, etc. So after the collection of sample, it has to be put in suitable transport media and sent to the laboratory as soon as possible with proper temperature condition. So now what is the importance of diagnosis of viral infection? So first point is to start with the antiviral drugs. So it is very important to diagnose the causative virus so that treatment or antiviral drugs can be started immediately so that patient will become normal within short period of time or as soon as possible. So it is also important that screening of the blood donors. So there are certain bloodborne pathogens which are transmitted from one person to another person through the blood. So it is very important to test the donor blood for these pathogens so that if the donor blood is positive for any bloodborne viruses like HIV, hepatitis B, hepatitis C, it should be discarded and it should not be transfused to the recipient of the blood. And third one is for surveillance purpose or for outbreak or epidemic investigation. So early detection of outbreak, that is sudden increase in cases of any particular type of infection. So it identifies the causative virus at the start of an outbreak so that we can take suitable or necessary precautionary measures to prevent the transmission of these infections. And also we can detect the genetic mutations in the virus so that we can identify the different variants of the viral infection. And the last part is to start post-exposure prophylaxis of antiretroviral drugs. So if any person unknowingly comes in contact with the infected individual through sexual contact or through splashes of the blood or through needle prick injury or through tattooing, etc., it is very important to start with the post-exposure prophylaxis as soon as possible. So here diagnosis of viral infection plays important role in the diagnosis so that essential antiretroviral drugs can be started with immediately. So now what are the different methods coming under laboratory diagnosis of viral infections? So first one is direct demonstration of the virus by microscopic methods. Second one is detection of viral antigens, detection of viral antibodies, specific antibody against the virus particle. These two are coming under serological diagnosis. And next one is molecular method to detect the specific viral genes, whether it is DNA or RNA. And the last one is isolation of virus, that is cultivation or growing of the virus. So first one is direct demonstration of the virus, which involves different methods like electron microscopy and the modification of electron microscopy, that is immune electron microscopy, which is more sensitive, then fluorescent microscopy, then light microscopy. So first one is electron microscopy. So we know that we cannot observe the viruses under light microscope. We have to use electron microscope because they are very small in size to observe under light microscope. So electron microscope is used, which allows the direct visualization of the virus particle based on their morphology, size or structural features. So here the specimen is mixed with the heavy metal stains like phosphotungstic acid, which increases the electron scattering and image contrast. So after the staining, we have to observe this under electron microscope. So different shapes of the viruses can be identified. through this method. Like for example, rabies virus has bullet shape, or rotavirus has cartwheel shaped structure, adenovirus has space vehicle shaped structure. So we can make out these different types of shapes. And next one is immunoelectron microscopy. So it is a more sensitive and more specific method compared to electron microscopy. So here we are mixing the sample with the virus specific antibody. So this antibody, it will bind to the virus particle and it will aggregate the virus particle, so that it enhances the identification and specificity of the test. After this sediment, this mixture is centrifuged and the sediment is negatively stained with the phosphotungstic acid and viewed under electron microscope. So next one is light microscopy. So this light microscopy is useful to detect the inclusion bodies by histopathological staining of tissue sections. So inclusion bodies are nothing but the structures which are seen with the different sizes, shape, location and staining properties, which can be demonstrated in the virus infected cells under light microscope. Here we are observing these inclusion bodies in the tissue sections of the infected host. For example, we can detect the Negri bodies in the brain biopsies of the rabies infected patients. The staining method used here is immunoperoxidase staining. Next one is fluorescent microscopy. Here the fluorescent dyes are used. So we have to mount the specimen on the slide. We have to stain with the specific antiviral antibody which is tagged with a fluorescent dye, and we have to observe this under fluorescent microscope. So it will be useful for rapid diagnosis of rabies virus antigen in the skin biopsy or influenza virus, etc. Next one is serological method, which involves antigen detection as well as antibody detection. Here the most common specimen used is serum, or other body fluids can also be used. So here for detection of antigen and antibody, there are various methods like ELISA, that is enzyme linked immunosorbent assay, direct immunofluorescence test, immunochromatography, that is rapid CART test, flow through assays, etc. So examples for antigen detection test involves detection of hepatitis B surface antigen and hepatitis B precore antigen for hepatitis B virus infection, and NS1 antigen for detection of dengue infection, P24 antigen of HIV, then rotavirus antigen detection, etc. So next one is detection of antibodies. So conventional methods which were previously used involves heterophile agglutination test, Paul-Bunel test for Epstein-Barr virus, or heme agglutination inhibition test, neutralization test, complement fixation test, etc. So newer techniques which are commonly being used nowadays involves ELISA, immunochromatography, flow through assay, enzyme linked fluorescence assay, etc. Examples for antibody detection test involves antibody against hepatitis B core antigen, antibody against hepatitis B surface antigen, antibody against hepatitis B precore antigen. So these all antibodies we can detect for the diagnosis of hepatitis B infection. Then we can also detect IgM or IgG antibody against dengue by ELISA. Then molecular method, which is the gold standard always. It is more sensitive, more specific, and it will yield quicker results than that of the any other types. So most common molecular method is polymerase chain reaction, that is PCR. So it involves 3 steps mainly, that is extraction of DNA, amplification, then detection. And there are different types of molecular methods like reverse transcriptase PCR, real-time PCR, multiplex PCR, biofire film array, etc. And last one is isolation of the virus by animal inoculation, embryonated egg inoculation, and tissue culture. So we all know that we cannot grow the virus in artificial cell-free culture media. we need the living cells for the growth of viruses. So we have to inoculate them into experimental animals or embryonated egg or tissue culture. So this isolation of virus is alone a important 5 or 3 marks question, which we will discuss in the next class. So laboratory diagnosis of viral infection as a whole, it can be asked for 5 marks. Thank you for watching. We will